Okay, so it's my pleasure to briefly introduce this month's speaker from EMBL, who is Nicola Gritti, who's joining us from EMBL Barcelona today. And so Nicola studied originally in Italy, Milan, where he did physics and finance, as I saw on his CV. And he then went on to do a PhD in quantitative biology in the Netherlands, and then went to do a postdoc with Jan Huysken's lab I think it was still in the US. And since now for 2 years or so, I believe you are now an image analysis specialist with the Barcelona unit and EMBL. And so today, I believe you're going to tell us a lot about high throughput analysis methods and various tools. So very much looking forward to this. So please, Nicola, take over.
So, well, first of all, thanks for having me here. As Robert said, my name is Nicola Gritti, and I'm an image analysis specialist at the Mesoscopic imaging facility, the MIF, at EMBL in Barcelona. Before starting the presentation on the method, I did not expect such a detailed introduction of myself, so I prepared a brief slide with a background and also an introduction of the team that I work in. So, well, as Robert said, I'm originally from Italy. I studied theoretical physics, and I did my master in the biophysics department, studying theoretical modeling of lymphocyte interactions in lymph nodes. And after this experience, I moved to the Netherlands at the Boehm Institute in Amsterdam, where I started working in experimental developmental biology, and in particular, I worked on post-embryonic development of C. elegans using a combination of microfluidics devices, time-lapse microscopy, and image analysis mostly. So after my PhD, I moved to the US in the lab of Jan Huysken, where I started working with light sheet microscopy, with infrared imaging, and in particular to use these kind of data sets to perform image restoration using deep learning techniques. In 2019, I moved to Barcelona at EMBL in the group of Vikas Trivedi, and most of the work that I will present today was developed during my postdoc time in his lab. And here I worked essentially doing some image analysis and image processing applied to the self-organizing properties of embryonic organoids. And very recently, so in November of this year, I moved to as an image analysis specialist in the Mesoscopic imaging facility with Jim Swagger, still at EMBL Barcelona. So this is a very recent transition. So the MIF, the Mesoscopic imaging facility, provides cutting-edge imaging to scientists within EMBL, but also outside of EMBL with collaborators. In particular, we focus on imaging large 3D samples that range in size from approximately 100 micrometers to the scale of a centimeter. So in the facility, we have spaces for instruments. We have wet lab space where we provide support to the users to perform the sample preparation and the mounting. And of course, we have offices, workstations, and virtual machines for the downstream image analysis and processing. So the team is composed by Jim, the head of the facility, Montse, and Gopi. And as I introduced myself briefly, also I was the last addition to the team in November of this year. So what does it mean to provide access to mesoscopic imaging for such a large range of scales, from micrometers to centimeters? The strategy of MIF is to provide a number of instruments that are complementary to each other. And these can be roughly categorized into 3 main types. So we have OPT, with which it's possible to perform tomographic reconstruction of 3D samples. And we have systems that can work both with fluorescence microscopy and transmitted light. We also have SPIM techniques, and in particular, we have 2 commercially available systems. One is the multi-view SPIM from Luxembourg, now Bruker, and which is a dual illumination and dual detection, and it can perform both live and clear sample imaging. We also have another system, the LS1, which is optimized for 4D imaging of organoids, so 3D plus time, with medium throughput, as it's possible to image up to 6 or 7 samples in the same imaging session. So on top of this, we have a hybrid system, which is OptiSpin. So it's a combination of OPT and light sheet microscopy, which is also can be used in live and clear configuration mode. Last year, finally, we also acquired a high content screening device, which is the Opera Phoenix from PerkinElmer. This is a spinning disk confocal microscope with which it's possible to image samples that are seeded in multi-well plates, so ideal for high throughput imaging. So altogether, these instruments allow us to look at large mesoscopic samples with different spatial temporal resolution. So these range from really highly resolved imaging of organisms and organs, such as the case of OPT and SPIM in the case of clear configuration. With the live sample SPIM, we can, of course, image biological processes at high time resolution. And finally, using high throughput imaging experiments, we can study the variability of some processes of interest. So the next challenge is, of course, to translate these beautiful images into numbers, so to quantify them, and to finally translate these into biological insights that can move forward the understanding of the underlying biology. So the two projects that I will present today tackle this question. And the first one is about quantification of images, in particular of organoids in high content screening systems. So this is more tailored to systems such as the Opera Phoenix. So in particular, we developed a software which we call MOrgAna. MOrgAna stands for Machine Learning Analysis of Organoids. So we've developed this software during, as I said, my postdoc time in the Trivedi lab, and we are now providing it as a tool to the increasing number of users of the MIF. So essentially, we designed this software based under the assumption that images of organoids can be acquired with a variety of different systems. So this can range from confocal systems to high content screening system, but also to simple benchtop microscopes. So these images of organoids can also acquire using different experimental designs. So organoids can, of course, be imaged over time, so perform time-lapse images. They can be imaged in replicates, multiple replicates, and then can be treated with different drugs, for example, so under different conditions. So what we developed is a software that naturally guides the users through two main steps, which are step one, the segmentation of the images, which is based on a machine learning approach, which is very simply divided into the training phase of the neural network, into the classification of the pixels inside the images, and to an important manual verification step to verify the fidelity of the masks generated. And next, these masks are then used to perform quantification, which is based on the experimental design, which, as I just mentioned, can be time-lapse, conditions, or replicates. And so the data can be visualized in the proper format depending on the design of the experiment. So the quantification provided by MOrgAna can be both morphometric analysis, so quantification of shapes, as well as fluorescence quantification. So for the morphometric analysis, we used MOrgAna to quantify the shape changes of, let me see if I can, okay, oops, sorry. The shape changes of during over time of the development of brain organoids. These were imaged in time-lapse microscopy on a confocal device, and the visual segmentation feedback is based on two modalities between which the user can choose from. And from the optimization of this training and segmentation algorithm, one reconstructs finally the final mask, which can be tuned by the user using some parameters, and in both cases can accurately detect the edges of the organoids. So this is an important step because you can imagine that over time, over the multiple days in which the organoids are imaged in these devices, these organoids can shed cells or debris can accumulate inside the wells. And so it's often very difficult to accurately detect the edges of these objects. So based on the final mask result, MOrgAna quantifies several morphological parameters, and here it's just a very short video to show you how it's possible with just a couple of clicks to, for instance, quantify, in this case, the area of a time-lapse dataset of brain organoids. And MOrgAna not only implements fairly simple morphometric parameters such as area, perimeter, form factors, and so on, but also we implemented more advanced morphometric parameters such as the log contribution elliptic Fourier analysis, which is essentially a way to describe the shape of organoids using consecutive ellipse fitting analysis. So in this figure, you can see this chymograph, which is for every time point in the time-lapse dataset, the contribution that every elliptical mode gives to the description of the shape of that organoid at that particular time. And the white dashed line represents the highest elliptical mode that is necessary to describe the shape of the organoid with 95% accuracy. So with this simple scheme, it's possible to determine, for instance, that at later stages where organoids grow in complex shapes, more ellipses as necessary to describe the shapes. In the same morphometric analysis context, we also showcase the power of MOrgAna in studying the patterning processes of pescoids. So pescoids are embryonic organoids that are derived from zebrafish embryonic stem cells. In this case, the pescoids were imaged on a simple benchtop stereoscope, and they were imaged under different conditions. So we have a control sample, and we have pescoids that were treated with a small molecule inhibitor for a particular signaling pathway at different time windows. So this experimental design led to images that contain multiple objects per image, which can be fed into MOrgAna, and every single object can be segmented out and treated as a separate object. So the importance of this slide is that with MOrgAna it's possible because pescoids grow typically in a band structure. So it is essentially important to compute the computational straightening of the objects inside the image. And this allows to not only look at area and major axis, but also look accurately at measures such as eccentricity. So in the last case that I wanted to show you is about fluorescence quantification. And in this case, we studied the patterning of the gastruloids. And gastruloids are embryonic organoids that are generated instead from mouse embryonic stem cells. In this case, gastruloids were seeded in a 96 well plate, and the images were acquired using a high content screening system, in particular the Opera Phoenix that I told you about earlier. So these are then imaged in high throughput during a long time. So in principle, often over several days, 5 or 6 days of development, and therefore they led to a very large amount of data. So in this case, the brain organoids were, the gastruloids were imaged both in bright field and in fluorescence, as they contain a gene expression marker for a particular gene that is important in the development of these organoids. So what we observed during a certain time window is that there is expression of this particular gene marker that is then polarized on one side of the gastruloid. And then in the next 24 hours, there is a dramatic elongation of this gastruloid over time in the direction where the pole is expressing this particular gene marker. So using the fluorescence quantification module of MOrgAna, which is built in parallel or on top of the morphological quantification module, we can then quantify these qualitative aspects that I just described. So in particular, for the first 24 hours, one can use the angular profile, so to study essentially the symmetry breaking event. So when is the polarization determined, and what is the preferential direction of polarization? While in the next 24 hours, one can look at anterior-posterior profile patterning to study the patterning of the elongation. And this in particular allowed us to look at the proportion of the gastruloids as it is elongating that is still expressing this gene marker. Okay, so these are the three examples that I wanted to show you. And MOrgAna, I just want to say that this is released as a Python package. So we have a GitHub page hosted by Lab Trivedi. And as common for Python packages, this can be installed on top of a pre-existing Python installation using the command pip if you install MOrgAna. And once installed, it can be run through a graphical user interface that allows users with limited coding experience to obtain results and quick visual feedback of their results, or through scripting to perform more high throughput analysis and integrate the results possibly with some custom written code. And in this case, we provide example scripts in the form of Jupyter notebooks. So with this solution, we provide an accessible tool to start applying machine learning algorithms to the analysis of high throughput data that are acquired with a wide range of devices. So for the second project that I wanted to talk about, I would like to introduce a new technique that I also started developing as a postdoc in the Trivedi lab, and that I'm now establishing as a tool for the users of the MIF. So in the zoo of the instruments that are available in the MIF, this solution is more tailored to high resolution 3D fluorescence microscopy, such as SPIM. And for instance, MIF users often use SPIM to visualize how individual cells move and behave inside a growing or developing organism or organoid or sample in general. So how cells differentiate in the spatial temporal context is typically represented using a Waddington diagram, in which cells are represented as balls that essentially rolls on this complicated landscape. So in this scheme, we have a time axis and we have a fate axis. And of course, here there is one crucial information that is missing and it's difficult to represent here, which is that of space. Of course, these cells are located somewhere inside the space within the sample. And for instance, this map doesn't take into account that even though these two cells that are very similar in terms of their fate, they might migrate actively inside the sample and end up in very different positions within the embryo. So essentially, to study how cells differentiate in the spatial temporal context, it is very important not only to look at their gene expression profile or their fate, but also to their temporal and spatial information. Of course, we know very well that it is very complicated within the same experiment to have access to all these three information at the same time. However, there are complementary techniques that allow us to focus on one or two of these aspects. So there are, of course, classical immunostaining techniques, or more recently mRNA staining techniques, such as single molecule fluorescence in situ hybridization or hybridization chain reaction, which give, of course, a lot of spatial information because every single cell inside the sample is visible in principle. And they also give a fair amount of information on the fate of these cells, assuming that the genes that are stained for are representative for a certain fate of the cells. Of course, because the samples in this case need to be fixed, there is no temporal information available. Next to this, of course, we have time-lapse microscopy imaging, with which it's possible to visualize with very high level of detail both the spatial and temporal information of single cells, in this case, inside a developing zebrafish embryo. In its simplest form, time-lapse microscopy does not allow, however, to look at the fate of these cells with very high detail. However, the fate of the cells, if we take a step back and we move out 1 second only from microscopy, can be visualized or can be defined even by single-cell RNA sequencing analysis, which is the gold standard to determine cell state and transcriptomic of the cell. So, of course, again, in its simplest form, single-cell RNA sequencing does not allow to look at temporal and spatial information because the samples need to be fixed and all the cells need to be dissociated. So the spatial information of the origin of the cell is completely lost. So this naturally brings us to the question, which is, can one combine these different experiments and gain some biological insights that would otherwise be inaccessible looking at only one of these datasets? So to answer this question, we propose to combine these techniques in an experimental pipeline that we call LinSeq for lineaging sequencing, in which first one would like to perform RNA staining with, in our case, HCR, so hybridization chain reaction, for some selected marker genes that are spatially distributed inside the sample to label some specific cell fate. This will give us a matrix for the XY position of all the cells inside the HCR image, as well as a matrix for the marker genes for each one of these HCR cells. Next, using some single-cell RNA sequencing data, these can be mapped onto the location of the HCR cells using the marker genes as anchors of the space. And this would allow us to generate a synthetic HCR cell transcriptomic profile. And finally, using the data that are the locations that are obtained from the HCR images, these can be mapped onto a data set from a live images, therefore generating again a synthetic transcriptomic profile of cells in a live sample. So to explore the possibility of designing such a pipeline, we focused first on the development of the zebrafish embryos, you can see in these cartoons. And this is mainly motivated by in two ways. So first, because it's a widely used model organism. And second, because single-cell RNA sequencing datasets are already available and they can be explored nicely. So of course, this pipeline in general, it is not specific to zebrafish embryos. So in the future, we would like, of course, to try this in other organisms as well. So then we set out to find some marker genes for some cell fate that cover a large part of the zebrafish embryo structure. And we decided to start from embryos at 12 hours post fertilization because a lot of the cell fates are already present at this stage. So we decided to focus on some head and tail and mid-trunk fates, such as the telencephalon, the heart primordium, the neural crest, diencephalon, and tail bud, which are represented here in images from the literature. So when performing HCR, we obtain a staining for these marker genes, which is quite similar from what we have seen in literature. And by using a ubiquitous nuclear marker, which is depicted here in gray, one can also visualize all the cells inside the embryo. Of course, at this point, one might ask, are 5 genes enough to look at all the structural complexity of the embryo? So the difficult part here is that, of course, when you do imaging, you are limited to a certain number of laser lines and filter sets. And so we managed to squeeze these 5 genes into the same imaging session by using the fact that, for instance, telencephalon and heart primordium are spatially separated. So even if they are in the same color, they can be post-processed and treated as separate channels. However, of course, one might think of adding more genes, and by performing different HCR panels, we decided to stain for different markers, which are depicted here, and thus generating a pool of 14 marker genes over different zebrafish embryos. So from these images, we then perform segmentation of single cells and eventually reconstructed a synthetic embryo. So the method is quite simple. So we perform different off-Gaussian analysis with local maxima. And this is GPU accelerated with this beautiful Python package from Robert Hasse. And with this, we could segment most of the cells inside the embryo. And then using the difference fluorescence channel, we could reconstruct or extract the fluorescence intensity profiles of every single cell inside the 3D space. So the next challenge is, of course, that of registration of different samples. Like when different fish are stained and treated in different days or different ways, there will be, for sure, some deformations or some registration problems in the imaging. So, for instance, here you can see that these cells from a single fish which was stained for this particular marker gene, when overlapped with the same fish, a different fish stained for the different marker gene, they overlap. very poorly, and they have a very bad deformations. So in this case, we decided to use another Python package, which is Vido, developed by Marco Musi at EMBL Barcelona, and perform local registration using single-cell warping, with which now it's possible through iterative local registration loops to segment this, to register these cells on top of each other, therefore generating a common 3D space of a synthetic embryo. So in this case, then we use this particular registration method on this 3D point cloud, essentially, to construct a synthetic embryo using nearest neighbor analysis. We can essentially map all the 14 marker genes onto a single synthetic zebrafish embryo. And now you're seeing these 14 marker genes as the embryo rotates. So, well, this solves the first part of the problem, to generate a large amount, large enough data set for HCR images and quantification of single cells analysis. The second part is to perform the mapping of a single cell RNA sequencing data set onto these locations of the cells from the HCR. So for that, of course, there are a lot of different approaches and a lot of different algorithms. And the one that we decided to use is NovoSpark, which uses in particular an optimal transport algorithm that essentially tries to minimize the distance between cells, both in 3D space and in the RNA space, as well as the discrepancy between the genes and the profiles of the cells that are embedded in the space. So, well, in other terms, this algorithm works under the assumption that cells that are closed in 3D space also share a similar transcription profile. The mathematical description of this is here. I will not go into the detail. I just want to mention that this parameter alpha is quite important and can be tuned in order to give more importance to the marker gene expression from the HCR, or to the assumption of the algorithm that cells that are closed in space also share a similar transcription profile. But essentially, when this minimization problem is solved, one obtains an optimal transport matrix, which is a matrix that has dimensions of number of locations by number of cells, which is then multiplied by the single cell RNA sequencing matrix to obtain a virtual HCR transcriptomic profile. So we decided to use, as I mentioned in the beginning, a dataset of single cell RNA sequencing dataset that was already published. And this is this dataset of 12 hours post-fertilization cells from zebrafish. It's a very rich dataset. It contains approximately 5,000 cells that are already classified and annotated in different tissue types in the 12 hours post-fertilization embryo. And then when we mapped our cells from HCR images purely based on our 14 marker genes, we observed that these cells essentially distribute on this 2D space. And then based on this distribution, one can, transfer the label of the clusters from the single cell RNA sequencing dataset to the HCR cells. So for instance, all these cells in this cloud at the top will be labeled as mesoderm cells, and all these ones on the left, epidermal cells, and so on and so forth. So to confirm the fidelity of the integration that we've just performed, we can also visualize expression of some of the marker genes for the tissue types, both in the single cell RNA sequencing dataset and in the HCR map dataset, and for each one of these clusters. So in this way, we can visualize if the integration worked and to see that these have at least a qualitative match. Of course, once the integration is done, the great advantage is that for each of these integrated cells, we also have the XYZ coordinates available as we obtain these from segmentation of the HCR images. So the next natural step is to visualize these cells in their 3D context and color code them according to the tissue type that they belong to. So here, for instance, you can see that in the head region here, we can see many of the ectodermal clusters. So the pink in the neural crest, the forebrain, the hindbrain, and so on. So yeah, this is the single cell RNA sequencing integration part. And so finally, to obtain also temporal information of these different cell fates, we turn to a time-lapse dataset that was generated by Gopi, as part of the MIF, during her time in Jan's lab. The beauty of this dataset is that by using a transgenic line in which 2 key marker genes were fluorescently labeled, Gopi was able to visualize the 3 main germ layers during the zebrafish development. So the ectoderm, the mesendoderm, and the endoderm. So, well, we looked at the cells that were segmented in this dataset, and using a very similar registration, local registration approach as I described earlier, for the HCR dataset onto the live dataset, we mapped the individual HCR cells onto these live cell positions and color code them by cell fate. So again, you can see on the left here, the color coded according to the live dataset, so the one generated by Gopi. And on the right, the synthetic dataset with the colors, all the different colors that are inherited essentially from the single cell RNA sequencing dataset. And so you can see, for example, that the big ectoderm region in the head here is now subclustered into hindbrain, midbrain, neural crest, and so on, and also some epidermal cells here on top. This is also evident in the bar plot here on the right, where the mesendoderm and the ectoderm cells now are reclustered into assigned more precise fates. So of course, the final step, this is just a single snapshot of the whole movie of 12 hours post-fertilization development of the fish. But of course, being this a time lapse, it's now possible to trace back these cells according to their color code and reconstruct a full single cell trajectory of the zebrafish embryo and observe how these different cell types emerge over time. So yeah, you will see now here most of these colors appearing by the end of the movie, and this can also be seen by the proportion of cell types that are present at the different time during the zebrafish development, where we start at the beginning with mostly endoderm, mesendoderm, and ectoderm cells, and we end up with many more cell types. So yeah, this brings me kind of to the end of the results part of my presentation. And as a take-home message, I would like to highlight that I have developed this during my postdoc time, and we are now providing it to the users of the MIF. And of course, it's possible to expand, and we're thinking of expanding all of this work in all possible directions. So for instance, one obvious thing that we did in MOrgAna was to limit ourselves to the analysis of 2D images. So one obvious future direction would be to expand this to 3D morphology analysis. For the lineaging sequencing project, we would like to try to improve the embedding with more marker genes beyond the 14 that we have used now. Also to use HCR with fish of different ages, because actually the single cell RNA sequencing dataset that I told you about in the beginning is much richer than what I showed you. So they actually did single cell RNA sequencing for fish at different ages. So we limited to the 12 hours post-fertilization, but in principle, it's possible to use also this information. And of course, there are also the possibility to expand this pipeline and apply it to other model organisms or organoids in the future. So finally, well, I'd like just to thank everybody, so people that I have worked with in the Trivedi lab. So in particular, of course, Vikas, Christina, Karim, David, and of course also all the people at the Mesoscopic imaging facility now and funding EMBL. And I'm happy now to take any question you might have.
Yeah, great presentation. Thank you very much, Nicola.
Thanks.
I'm sure there are questions, so if you have them, please raise your hand. Maybe while we're waiting for a first question, I was very interested in the first part of your talk, the MOrgAna tool, and maybe you can tell me again to which type of microscope modalities have you already applied it. So I think I realized it was a lot of confocal fluorescence based. But then in principle, you also try other?
So we use it for standard confocal devices, high content screening devices, and benchtop devices. So in the case of benchtop stereoscope, of course, it was essentially limited to the analysis of bright field images. And in the two other cases, there was also the fluorescence quantification involved in it. So we have limited this to these three systems because, well, I think that at least, in our unit, these are the three systems that are mostly used to look at organoids because they allow to, perform high throughput analysis, especially in the case of high content and benchtop.
Mm-hmm. And so can you maybe also comment a bit on how large the data sets can be? That because we're sort of interested in maybe, applying this to volumetric video data, which can be then sort of scale, easily go up to terabyte scale.
Exactly. So especially in the case of the Gastruloids slide that I showed. So if you think of even acquiring just a single focal plane on a high content screening device every 5 minutes for 5 days, it's of a 96 well plate. It's essentially generating up to 3-4 terabytes of data, and so it is definitely something that one has to keep in mind, of course. I think with MOrgAna, we try to, for this large data set, we always encourage to use the Jupyter notebooks that we provide, of course, because it's possible to just run them for the whole time of the analysis without bothering. And we also optimize for speed for the analysis. Like there are some parallelization processes and some GPU accelerated parts inside the code. So we found that for a 24 hours data set, because usually organoids on the high content screening device are imaged in 24 hours chunks because one needs to change the media, for example. For a single 24 hours window, it takes approximately 4 to 5 hours to analyze all the images inside every single well at every time point.
Yeah, sounds good. Thanks.
Thanks.
Do we have any other questions? Yeah, Ulrike.
So I have actually a couple of questions. So first of all, thank you very much for your nice presentation. That was actually very insightful. It's like amazing to see which tools have been developed and which is also so nicely in sync then with like the imaging technologies which you have at the center, and that you can immediately actually provide that service. But so my first question would actually go more into the direction of that is actually, I mean, this must have been super nice to see that what you've developed as a postdoc can right now further develop, but also be maintained and even established as tools within that center, right? I mean, could you maybe say a few words? How can I say about the importance, more or less, of having the chance to actually establish these type of tools, because I do think sometimes it's really so that during a postdoc, one develops a tool that's then there for a specific purpose, but not necessarily accessible to the entire community. And right now, I actually do think that you are in a great position, that you have kind of the security of that center, but you, in order to actually guarantee that this open source software tool is accessible to the world. I mean, maybe just a few words about how you see this, and what's also the plan long term with newer software tools, like how you plan to also distribute this within the center and from there to the world.
Yeah. Well, I think that's a great point. I mean, I think it's extremely important, especially during the postdoc time, as you mentioned. People tend to develop their tool, and unfortunately, very often it's the case that when the postdoc leaves or the project is done, the tools are kind of, they fall into hidden space, no? So I think in my case, I was extremely lucky because I did, essentially all of my PhD and the 2 postdocs I did working on, mostly image analysis. And so it was kind of an ideal continuation for me. It was not, it was not a single project that I worked on. No, so it was definitely helpful in my case to work in an environment where people in Trivedi lab work a lot with people in the mesoscopic imaging facility. So there was already in my postdoc time in Vicas's lab a lot of interaction with the people there. Of course, both of these projects started developed for people in the lab where I was, and then we realized slowly that these could be useful tools also for people in the unit and then to the outside. So now that I am in this position, well, these are kind of projects that we have developed and we want to continue. So I'm in the ideal situation to continue this now. So yeah, I think it's an extremely important part of the work of a postdoc to maintain these tools that are developed.
I couldn't agree more. thanks for pushing this, and also like that you've set up these Jupyter notebooks with example code and so on and so forth. I mean, that's amazing, because sometimes people, in order for them to use and apply these things to their own data sets, I mean, they need to sometimes be guided through this a bit better. And I think right now there are tools out there like Jupyter notebooks and so on and so forth to actually do this. And I think if everyone in the community of the open software developers uses these tools in order to make sure that the users then can quite quickly pick up on these tools, that would be amazing. And I think Hughes is a great example of this. Yeah. Thank you.
Yeah, no, I couldn't agree more. I mean, now we have a lot of tools that will allow us to share our results, our tools, and our softwares, and make them accessible to everybody. So,
I also fully agree. Yeah, Sinem, please.
I was actually just going to continue on that. I mean, so I think these are great tools for organoids, but can some of them be also just leveraged for other types of samples, like tissue blocks, 2D, 3D? And, like how much more input needs to be added to actually use them for other types of samples too?
Mm-hmm. I think the two tools that I showed you, they started in the origin not actually being like organoid tools. Of course, MOrgAna, yes. The other one, not really, in the sense that I could see some things that needs to be extended into the LinSeq to make it work in the organoid case. And one thing that we will have to think about if we want to do it is that, of course, organoid development is much more variable than embryos development. So we started from zebrafish embryo, which is developing in a very stereotypical manner. So being able to map cells from HCR from single cell datasets to HCR to live samples. These are three different samples of the same organisms or organoids, and you have to rely on the fact that these cell types exist in all three datasets, and they are more or less structured in the same way in the space. And this is not necessarily the case always for organoids. So I can see how this could be something that we would have to think about if we want to apply to organoids. In the case of MOrgAna, I think, we designed it definitely for organoids. I think that there is no, there is no limitation in it to use it for tissues or other types of samples, because it's essentially a morphometric analysis pipeline, and as long as you have an image where your sample is inside that image, fully inside the image, and you want to quantify area, perimeter, shape changes, and so on, and as long as you have a fluorescence intensity channel where you want to quantify, I don't see. I mean, it's very general, so I could imagine that it could be well applied to. Yeah, the only limitation that I see there is the 2D, 3D case. So for now, it's limited to 2D. That's definitely the case, because we wanted to start using this tool quickly. So because we had a lot of data that we acquired, and this project actually started during lockdown. So that's when we generated, and we ended up with a lot of data that needed to be analyzed. And these were data from the Opera Phoenix, so from high content screening device with 2D datasets. So we limited to this. We decided to limit it to this.
Thank you.
Great. Are there any other questions? Yeah, Ulrike again.
So I actually have another question about the logistic of the data analysis, processing, and storage logistics of your center. So right now you have tools which actually produce a massive amount of data, right? And then these tools, because your unit is like a service unit within EMBL, which is accessible, I guess, to all EMBL sites within Europe, right? So people come over with their samples and they do their measurements there, and then they produce the data. So the big question is, do you do the analysis there? Do you do this remote? How do they take their data with them? How long would you store the data, and so on and so forth? And which way have you found which would be the best way to also transfer the data? Meaning, are you using Amazon storage capabilities? Like, could you give us a little bit more insights about, as I said, the entire data management from, as I said, the acquiring of the data over the storage, analysis, and also how long would you personally, because right now you are, I think, then more or less the only person for the responsible for the development of data analysis, but I guess there are more members on your team who can help with that. But I can imagine that there must be a ton of projects. So you are also, beside of doing the development, also occupied with helping users with the quantification. If you could give us a little bit more insights into the entire data flow.
Yeah, well, this is a super important question, yes. So, well, I think there are two important things. I mean, with users of the MIF that are within EMBL, of course, we have a server storage system that can be accessed by anybody inside EMBL. So typically, of course, the flow is we acquire images and either stream that directly to the server, and they can be accessed basically real time as they are acquired by the users that is sitting in the office or even at home through VPN, of course. This is one case. So this is the case. This is the route that I think I would prefer, because, well, it's transferring of terabytes of data is already a limitation, becoming a limitation for these big data sets. No? So I mean, transferring I don't know, 10 terabytes of mouse cleared sample. It's several hours, and that also is a lot of money essentially. So I would prefer essentially to use, and this is actually what we do with everything that we have used MOrgAna for, because it was everything internal to EMBL. So all the analysis was performed real time as the images were acquired, more or less. For users that are external, they don't have access to this server facility. So typically, I think we ask them to take the data. So there is, of course, the possibility to store the data on the local machines there, and then they can be taken either by hard drives or external hard drives or directly to their networks from the center there. So for the part of how to handle the image processing and data analysis part, yes, there are a lot of users and many people have different projects. And yes, sometimes I can imagine, well, I am very new to the MIF, so I have not had the luck to be involved in thousands of projects and find myself completely overwhelmed. but yeah, definitely I can see how in the future there will be a lot. Well, there are already a lot of people that do imaging with different, very wildly different samples. I think the key there is to guide the users in the direction of the tools that is best suited to their sample. Like, one shouldn't necessarily need to sell MOrgAna if I know that MOrgAna is not useful for them. So it's something. So this connects a little bit to the discussion we were having a minute ago. No? So nowadays it's so easy to share softwares and make them user-friendly. Well, if we want, that with the right guidance, in principle, we would like to design these tools to make the users like kind of independent. No, I think there is always, well, in this kind of interactions between users and support staff. There is always a two ways interaction. So one needs to be willing to provide tools that are available and user friendly, but also on the other hand needs to be willing to learn how to use them and to spend some time and not ask somebody that has done it or knows it to perform everything. No? So this is my personal opinion. But
no, thank you for these remarks. That was very useful. Thank you.
Okay, so do we have some more questions? I don't see any, and if not, then yeah, maybe I hand over back to Ulrike for some last announcements, and then we can transition to maybe the more more informal session where we can ask questions, right?
Yeah, perfect. So before we dive into the Meet the Speaker session, I maybe like just a few announcements for the next year. Or first of all, I have to say that we reached the end of the year, so that's the 12th talk this year. Or there were even more talks actually. But anyway, thank you very much everyone for joining us, and I hope you enjoyed all the sessions this year. And we, all the hosts and organizers, are very much looking forward to welcoming you back in January with very exciting sessions. So the first talk in January, unfortunately by Chen Wang, was again postponed, but we are looking for a replacement speaker there. But at the end of January, we actually have a very interesting session by Karl Svoboda. So Karl Svoboda was a group leader here at Janelia, but he recently joined the Allen Institute in Seattle and is right now, I think, spearheading there also a department about, I think, dynamics of neurons and the brain and so on and so forth. I think this will also be super insightful. I don't know exactly what he's reporting on. Maybe he will just give an overview about what he has done so far, even here at Janelia, but I'm pretty sure that he will also give us very brief insights into what his new plans at the Allen Institute are. And I'm pretty sure this will be actually super exciting. So again, thank you very much, everyone, for accompanying us until December, from January until December. And we really very much hope that you enjoyed the sessions. And please also feel free to give us, meaning the host at the EMBL, but also me, feedback about what you like about the Janelia and EMBL bioimaging seminar series, what we could maybe improve, or even also speaker suggestions for upcoming series, for upcoming talks within the next year. But with that, as I said, I wish you actually a nice Christmas, nicely themed, and also a nice, in Germany, we would say, smooth slide into the new year. I don't know if there's an English expression for this, I don't know, but I wish you a happy new year. And yeah, and see you in January then. Bye-bye. And as I said, for those who would like to stick in the Meet the Speaker session, please stay on this channel. Otherwise, see you in January. Bye-bye.